Good afternoon. I'd like to welcome you to the first of the spring PBD Science and Technology Talks. Our speaker today is going to be very interesting. It's staff scientist Caroline Ajo-Franklin, who works at the Molecular Foundry. She's also affiliated with the PBD. Before she gets started on her talk, I have just a couple of announcements to make. We have the Spring Science and Tech Talk schedule. It's available on the table out there. And for those of you who are listening who are postdocs or working with postdocs, we have a very interesting event planned on Thursday. It's going to be here in the Building 50 auditorium beginning at 1 o'clock. It's the kickoff event of the PBD Postdoc Network, featuring a dynamic speaker named Erna Grass. Now, to our speaker this afternoon, Caroline Ajo-Franklin is going to tell us about her research on reprogramming the respiratory pathways of organisms. So I'm going to turn it over to you, Caroline. Welcome.
Thank you. Thank you. Okay. So I am very happy to be here this afternoon, partially because I am now affiliated with PDB, so I hope that this will be an opportunity for me to meet you guys as well as introduce my research and get more involved. So what I'm going to be telling you about today is the work in my laboratory over the last couple of years on trying to harness the ingenuity of nature over the last 4 billion years and to use the systems and proteins that nature has evolved and transplant that into new organisms so that we can actually engineer organisms as if they were materials and change their material properties, such as conductivity. So that's what I'd like to tell you about today. So my laboratory really is focused on the idea, actually, of functionally interfacing living systems with inorganic materials. So really at the interface between living systems and non-living systems. And there's two major focuses in my group. The first one, which I'll spend most of my time talking about today, is the idea of electrically interfacing these two systems so that you can enable the bidirectional flow of electrons between living systems and non-living systems. The second area that, if we have time, I'll get to talk about is basically the idea of using living organisms to remodel materials, to make and break the bonds that form minerals. Okay. So at both of these, why do we do this? Well, the reason we do this is very well aligned with the laboratory's goals. We're really interested in using these types of approaches for carbon neutral energy. So in the case of sort of electrifying bacteria, we're really interested in the idea of, can we take solar energy, add it to CO2, and then use microorganisms as a catalyst to make fuels? So a solar to fuel route. Alternatively, in the case of the mineral, creating minerals and sculpting them, what we're really interested in doing is taking CO2 in the gaseous form and converting it into the solid form, so into carbonates, which you can just simply haul off and store. So there's a much safer way of storing CO2 over the long term. Okay. So let me just go ahead and dive in and start talking about electrical connections. And just to be clear, since this is not a humongous audience, if you ever have questions, just feel free to interrupt me. Okay. So what really motivates this is actually partially the expertise of JBay and JCAP here at LBL. So the real strong points of JBay, of making biofuels from biomass, is that you can make very complex fuels that are readily adapted to our existing infrastructure. However, the real problem with this approach is, as we know, is that you have to use water and land that can basically compete with food production. So that's a serious concern in scaling this up on a global scale. And basically, the big problem is that solar harvesting from natural photosynthesis is pretty inefficient. So those attributes have been what has fueled, in many ways, the idea of doing artificial photosynthesis, where we could actually get away from the water use issues because we're not making biomass. Instead, we're using light directly, solar energy directly with CO2 to create fuels. And we can do it much more efficiently because solar harvesting by inorganic materials is much more efficient. The real problem is that at this point in time, the diversity of fuels that we could make is pretty limited. So what one emerging idea is the idea of electrosynthesis. And the goal of this approach is to really bridge the two complementary attributes of the J-Bay and the J-Cap approaches. So to actually take solar energy, harvest it using inorganic materials, do water splitting reactions in the inorganic materials, and then shuttle electrons, electricity, and CO2 into a microorganism and use it as a catalyst to make complex fuels. And the goal here is that by this approach, you would be able to capture the complex fuels that J-Bay can make and also take advantage of the high-efficiency solar capture that J-Cap can really manage. So how do you actually do this? And how is this being done? So currently, there are two basic approaches in the literature. The first one is from Derek Lovely's group, in which they use acetogens that fix CO2 and also take electrons from an electrode and use this to make pretty simple fuels, so things like butanol. And the basic challenge with this approach is that, as you can see, there's not a dense array of these microorganisms on this electrode material. So the flux of electrons coming in is really low. So if you want to make a fuel, you want a lot of CO2 and a lot of electrons in, and that's going to help you drive making a fuel very efficiently. On the other sort of main technology has been out of Jim Lau's group at UCLA. And what he's done is actually genetically modify Ralstonia eutropha, which normally grows off of CO2 and hydrogen, using the hydrogen as a source of electrons to make fuels such as butanol again. And the real challenge with this approach is that they're using an incredibly high overpotential to make the hydrogen from an electrode. So the overpotential is greater than it needs to be by about 3 volts. So that actually makes the efficiency of the process about about 4 times less efficient than it really needs to be. Okay. So if the real problem is basically getting electrons into a cell that also fixes CO2, there are a couple other approaches to try and enhance electron fluxes into the organism. So, for example, Derek Lovely's group is trying to find better microbes. So microbes that naturally take in more electrons and still fix CO2. The other approach is to actually use mediators, so use redox active molecules that allow electrons to come into the cell. So here's an example of a redox mediator from Guy Bazan's group that actually can move electrons across the membrane. Okay. So the problem with this approach really is that you're limited to what's in nature. So you're hoping that nature has optimized something that fixes CO2 and takes in hydrogen, or it takes in another electron acceptor. And that, as an engineer, I kind of find a little limited. I'd like to go beyond what nature has made. In the case of using soluble mediators, the big problem is you can get electrons in, but you have no way of controlling where they're going. So it's kind of like just shotgunning them in, and you have no way of making sure that they get into the biosynthetic pathways that you really need. So what our approach has been is to take an organism that has already done a fantastic job at developing a system for moving electrons in and out of the cell, and then to actually transplant that into a natural system that fixes CO2. So that basically what you're doing is taking the best CO2 fixer and the best electron transfer organism and slamming them together. So that's our vision for where to make progress in the electrofuels system. Doing this is no simple challenge, so what we've started out doing is using E. coli as a test bed, because we think that if we can't transfer this ability to move electrons in and out from these native organisms into E. coli, we're going to have a lot harder time doing it in organisms that are not as genetically tractable and don't have as many genetic tools. So what we'd really like, our wish list is, can we create a modular and molecularly defined route for electrons to enter an organism and exit it? Of course, we'd like that to be able to reduce or oxidize an electrode. We want quantitative control. We want it to be fast, efficient. And we want to also try and be able to direct a surface attachment. Okay. So the system that we were inspired by is the MTR system of my favorite bacteria in the world, Shigella odenisis MR1, and its friends call it Shiwi. So what Shiwi does is it actually has evolved in environments where it doesn't have oxygen or other terminal electron acceptors to respire on. So what it has learned to do is to respire on solid minerals. So you can actually see, if you look carefully, there's a little gap right here that's about this bacteria size. What that results from is that this bacteria has actually literally breathed iron, reduced it, and then the soluble iron dissolves away, leaving a little pit. So that's basically where it left its breath behind. So how does it do this? Well, what it does is it's got this very extraordinary electron transfer pathway that spans the two membranes of this gram-negative bacteria. So what you do is you take a sugar, and you oxidize that sugar to acetate and CO2, and the electrons from that sugar end up on this metaquinone. Then the electrons from the metaquinone are passed through this MTR pathway. And what this pathway is composed of is a series of electron transfer proteins, first in the inner membrane, in the periplasm, and in the outer membrane. They can literally carry these electrons like it was a little baton, just passing them from one protein to the next. When they do this, they can both reduce the solid iron on the outside of the surface, here, or they can reduce an electrode. Also, if there's chelated iron, you can actually get the chelated iron to be reduced in the periplasm. So one of the amazing things is that this pathway is also reversible. So if you switch the bias of the electrode, so now the reaction becomes downhill in energy, you can actually send in electrons into the cell. And we know that we can reach the quinol, and it's hypothesized that you could eventually get to NADH, which is or NADH or NADPH, which would be a reducing equivalence for making biofuels. So not only do I love this pathway because it's reversible and it spans 20 nanometers, but because the proteins involved in this pathway are so awesome. So this is a homologue of our MTRC. They contain 10 hemes. And you can actually see these hemes in red. They actually provide a stepping stone for electrons to move through the protein. So you can almost just see where the electrons could flow through this protein. So they're awesome, but they're awesomely complicated and thus hard to make. So when we set out to transfer this pathway into E. coli, we decided to give ourselves a break, and instead of doing something that was 4 times harder than anything anyone had ever done, was to do something only 3 times harder by just incorporating these 3 proteins into E. coli. And we knew that there was significant homology between this protein, NAPC, and the partner in Shigella, so we thought we could get away with that. Okay. So what we did, and by we, I mean actually Heather Jensen, who is a graduate student in my group, who is getting ready to graduate, so she's on the lookout for postdocs. So what she did is actually made two plasmids. One that contains the cofactors required to make these heme-containing proteins, to be able to insert the hemes into the protein molecule. And then another plasmid that carried either the mTRA molecule, just the first step in the pathway, or a plasmid that contained mTRC, A, and B, so the last three parts of the pathway. And what she thought would happen is if just making the mTRA strain, you should be able to reduce soluble iron, because that's how far the pathway goes. But if you make the complete strain, the mTRCAB strain, she thought she might be able to reduce solid iron. And she was able actually to express these proteins, and hopefully what you can see is that these are nice red pellets. That's the color that cytochromes are red, so you can easily see that they're being produced. And likewise in the mTRCAB case. So when she threw in iron citrate into, thank you, that becomes much easier. Yeah, now you can see it's red. So when you threw in iron citrate to these E. coli's, you've removed any other terminal electron acceptor. There's no oxygen present. What you see is the mTRA strain can reduce that iron 3 into iron 2 over the course of several days. And it does so about 8 times faster than the wild-type strain. Then we threw in actually little nanoparticles of iron oxide. So my colleague calls these, Snicker-size iron oxide for the bacteria to eat. So again, what we see over the course of about 2 weeks is that the wild type in the mTRA strain don't create much iron 2. They don't reduce much iron 2. But the complete strain is able to produce a substantially larger amount of iron 2. So what that tells us is that this pathway is actually able to function in E. coli. So what we know is that we've got a modular system. It's got a molecularly defined route. And so what we wanted to do is address these next questions. So we had made mTRCAB E. coli, or as I like to call it, E. coli, where the E is an electron. But when we compared it to Shigella, what we found is the rate of electron transfer was about 3 orders of magnitude slower than Shigella. And if you looked at the coulombic efficiency, so how many electrons you provided to the cell versus how many ended up on iron, compared to Shigella, or pretty much by any metric, it was just abysmal. So we sat back and pretended that we were engineers and asked the question, what are the big quantitative differences between these two systems? And a couple jumped out at us. First of all, if we look at the terminal electron acceptor, in our E. coli, we were only getting about 70 molecules per cell, whereas in the native organism, it was increased by a factor of, it was 1,000 times higher. The other thing that jumped out at us was that we were using an E. coli protein to feed into a Shigella set of proteins. So while it worked, there's no reason to think that that's necessarily an optimized interface. So we thought, well, maybe we should also consider changing that protein to the native Shigella. So we took these two approaches in parallel, and I'll first tell you about our efforts to bump up this mTRC. So when we looked at our electric E. coli, what we saw was, yes, they reduce iron, but they also looked sick. So for example, if you grow wild type or strains just containing the CCM plasmid, they grow up faster than if you've got the mTRCAB plasmid. And this may look like a small difference, but it's actually about a 30% difference in growth rate. So it's pretty substantial. The other thing that we noticed is all of this time we had been expressing these proteins at very low levels. So we had just allowed basal transcription, so very low-level transcription, to make these cytochromes. And the reason is because if we transcriptionally upregulated it, so we told the cell, make more of these proteins, the cell pellets got less red, making us think that it was making less cytochromes when we tried to ask it to make more. So the third piece of evidence suggesting that our system was just not in tip-top shape is that you can actually check the morphology of the cells by looking at forward scatter and side scatter on a flow cytometer. And what we saw is that the cells, when we were incorporating this full pathway, had greater forward and side scatter. What that means is the cells are bigger and they've got more granularity. And like with humans, this is when cells get bigger and more granular, that's a sign of stress. Just like with humans, you can see people getting stressed out get bigger. So, at least that's what happens to me. So, what we thought was that we're asking the cell to make 3 of these really complex proteins. And I'll just walk you through exactly what we're asking it to do. So we're making not only the cytochromes that we need, that are the electron transfer proteins, we need to make the biogenesis machinery. And there are 8 transmembrane proteins that are associated with this biogenesis machinery. That's a lot of proteins. So not only they're being transcribed, they have to be secreted through the inner membrane into the periplasm. And then this biogenesis machinery will take heme that has been synthesized from the cell and combine that with these apopolypeptides to make the complete protein. The thing is that there's a lot of ways that this can go wrong. So you can if you have too many polypeptides that need to go through this sex system, you can actually block it and eventually cause cell death. But even with lower level, you can basically mess up the entire proteome of this inner membrane. The other thing is, even if you secrete it into the periplasm, these polypeptides, unless they get heme incorporated, will just get chewed up. So these polypeptides, if they don't get 10 hemes in before a protease sees them, they will just get degraded. So what our hypothesis is that this is really a very dynamic process, and we've got to keep the rate of transcription in line with these other rates, or else we could be causing significant changes in the intermembrane proteome, or we can be making protein but not actually making any cytochromes. So this is where a collaboration with BioFab really came into play. So we wanted to be able to basically tune the transcriptional levels of both the biogenesis machinery we were making and the cytochromes that we were making. So we needed a way to do that. Fortunately, the BioFab had been making a series of a promoter library that basically allowed you to tune, have a promoter strength that spanned 4 orders of magnitude in a quantitative and well-defined way. So that gave us one way of actually being able to turn that knob and know how we were turning that knob. We also needed a way to do that for the cytochromes. So what we turned to is we basically found this strain, which has lower levels of the RNA polymerase that transcribes that works on this T7 promoter. So what that allowed us to do is actually show that in this system, you can change the amount of IPTG and change the promoter activity, again, by about 3.5 orders of magnitude. So now I've got 2 orthogonal knobs to actually turn up and turn down the transcription of these proteins. So what we did was actually create a whole bunch of strains and grow them up under different conditions, 196 conditions, I think, where we're tuning both the transcript level of the biogenesis machinery and of the cytochromes. And this is what our experiments look like. We grew up the cells, then we took a small aliquot, spun them down into these 96-well plates, and then we literally took the plate and put it on a flat band scanner and imaged it. And what we were looking for were two things. First of all, we're looking for cytochrome C expression. We're looking for red cells. So you can actually look and see here, these cells aren't very white, or aren't very red, but as you come over, they become redder. So you can actually just visually see which ones are producing the most cytochrome. The other thing that, if you look very carefully, you can see is that these cell pellets are different sizes. And so what that was telling us is not only are we affecting how much cytochromes we're making, we're affecting the biomass that we can make. And we saw that actually the physical size of these, the apparent surface area of this, was actually linearly correlated with the biomass. So this gave us a really fast and easy way of looking at those two parameters. So how healthy are the cells and how many cytochromes they're making? So first of all, how healthy are they? So what you can see as we scan the promoter strength for the CCM and the MTR-CAB is that actually, so white is high biomass and black is not high, is lower biomass. And it can go down by about 35%. So you see a pretty dramatic drop as you induce these genes, and a somewhat lesser so in the other direction. The other thing that you can see is you can look at the redness, and what you see is that there's just this very localized region in which the cells are most intensely red. So if you go too high in either the biogenesis machinery or making the cytochromes, you'll actually end up, again, with less cytochromes. Okay. So the first thing we wanted to do is just quantitatively confirm this. Flat band scanners are fast, but they're hardly quantitative. And we saw exactly the same trend as we. So what I'm doing is just taking a 2D slice through in this direction as you change CCM promoter strength. And what you see is, again, you see this steady increase and a drop-off. And that exactly correlates with what we saw in the plates. Likewise, when we scanned across the MTR promoter activity, we saw an increase and a pretty abrupt decrease. So we confirmed what we saw in the plates, and so we had this question: why does making more biogenesis machinery or making more polypeptides of the cytochrome give you less? So we set out to answer that question. For the CCM, it turned out to be a pretty straightforward answer. So what we did is just measured the amount of CCME, one of the subunits in this biogenesis machinery, and we saw that as you go up in promoter activity, you increase the amount of protein present. So this is a Western plot against CCME. But after a point, you suddenly lose a lot of that CCME. And what we think is going on is that basically you're blocking the secretion pathway. So you're making more transcripts, but they're not getting secreted into this region, and so they're being degraded. So that kind of sets an upper limit of how much biogenesis machinery we can make. We thought the same thing might be happening with our cytochromes, which also have to be secreted. And what we saw was that was not the case. When you went from this region of high cytochrome to this region of much lower, the levels of CCME, the levels of biogenesis machinery were the same. And you can quantitate that, and it's very clear. What we then did is actually calculated the efficiency of which we were making these cytochromes. So what I've done is shown you the amount of the MTR proteins we're making versus how strong the promoter is, and plotting it, again, as a function of the promoter strength. And what you see is the efficiency drops by about a factor of 300 as we make the promoter stronger and stronger. So this is kind of very much like Sisyphus. we're trying to make more cytochromes, and the rock is getting heavier and heavier the harder we push on it. So what we've come to think is happening is that basically these apoproteins are all competing for the same site on this biogenesis machinery. And this biogenesis machinery is from E. coli. It's used to only putting in at most 5 hemes in a protein. We're suddenly asking it to do 10. And we think that basically the enzyme is not processive enough to insert all 10 in one go. And so it releases a partially matured protein, which then gets degraded. So that would actually explain why you see this drop in efficiency as you produce more and more of these polypeptides. So that's our working hypothesis. Okay, so what we've done basically is now create essentially a phase diagram that allows us to explain why we get high cytochrome C in this middle region. Okay, but let us return to the question of how do we get better function? I've showed you how we can get more cytochrome, but we're really interested in function. Okay. So what we did is basically looked at both the biomass and the cytochrome C, how that changed with these 2 promoter strengths, and picked out 2 strains. One that basically had the most cytochrome C, so the most number of complexes that could move electrons. Then we also picked out a strain that had, about half of that number, but the biomass, the change in biomass was not nearly as impacted as in this strain. Okay. So we called those the most cytochrome C strain and the least perturbed cytochrome C strain. So we then went to Lars Angant's lab at Cornell, and his graduate student Michaela worked with us in inoculating these cells into 3-electrode electrochemical reactor. And what we were hoping to see is basically if we could feed these cells lactate, they would oxidize the lactate into acetate and CO2, and then move the electrons to an anode, where they could then, at the cathode, actually reduce protons into water. Okay. So what we found was that our original strain, the one we had started with, basically showed very little, showed no significant current relative to its control, where we just had the CCM genes. In contrast, this least perturbed strain showed substantially more current than any of the other strains, and about 10 times more than its control. And pretty surprisingly, the strain that had the most cytochromes, the most proteins that were designed to reduce the anode, showed the worst performance. So they couldn't deliver current to the anode at all. And these measurements are done on the per-cell basis. So we know that we're really comparing the same number of cells in all of these cases. Sort of backing that up, we looked at the amount of lactate which was consumed. And again, we saw the same trend. Basically, these strains were not very significant in degrading lactate, whereas with this least perturbed strain, we saw a significant jump in the amount of lactate. So what that told us was that these cells were actually using lactate to deliver those electrons to the electrode, which is all consistent with this idea of how the electrons are getting there. Okay, so that's really exciting to me because we've basically taken E. coli and shown that you can actually engineer E. coli to move electrons in and out, out of the organism. What's also somewhat daunting is it tells you that basically sort of you have to engineer the entire system at once. You can't just think that making the most cytochromes, the most tunnels, as you will, out of the cell is the best approach. You actually have to look at how healthy the cells are. And we're really interested in diving down and figuring out exactly what it is about this least perturbed strain that makes it perform so much better. Okay. So I also mentioned that the other thing that we're really interested in doing concurrently is basically looking at changing the electron donor to, so basically taking an E. coli protein and turning it into Shewanella protein so that the entire pathway is composed of the same proteins, or same proteins from the same organism. So this is what Heather did, is she actually went ahead and first created just a Syme-A MTRA strain and compared that to our MTRA strain, where you're looking at soluble iron reduction. And what you see is that basically the Syme-A MTRA really makes a huge difference. And this change is about, I want to say, 7-fold in terms of rate. What was also really interesting that she noticed is that this helped the cells survive longer. So just to kind of give you perspective, what we've done in these experiments is given E. coli all the food that it can eat, but we've removed anything that it can respire off of. So it's like we've removed all the oxygen out of the room. And so the only way it can survive is if it reduces this iron. And so what you would expect is that basically if it reduces iron better, it survives longer. And that's exactly what we see with the Syme-A MTR CAB, or Syme-A MTR strain. Survives quite a bit longer than the other strains. The next thing that she did is actually figured out a way to express all of these 4 proteins in a single strain. And this was really guided by her previous work in seeing how being able to tune the transcriptional levels of these cytochromes quite a bit. And what we can find is, even though this gel isn't the best in the world, that we can make Syme-A, MTRA, and MTRC in a single strain, whereas in the previous strain, you didn't see any Syme-A. So as we kind of had hoped, what we see is that if you go from the wild-type E. coli to the MTR-CAB to the Syme-A MTR-CAB, you see a steady increase in the ability to reduce iron. So that's telling us that, yeah, Syme-A also helps with solid iron reduction. The other thing that she's noticed, and we haven't yet fully explained, is that while it does help with solid iron reduction, it doesn't really increase survival that much. So we're really interested in figuring out why that is. Okay. So just to summarize where we are with this, what we've done is take a bacteria, E. coli, that couldn't respire off of iron or an electrode, added the right proteins, and then shown that you can reduce both solid iron or an anode. And what's really, I think, exciting about this is that this is totally modular and controllable. We're using the language of genes, so it should be transferable to any organism. And we think, especially when we're seeing this correlation between survival and function, we think that this is actually a very evolvable system. We've also learned that these cytochromes, their methods of maturation are much more complex than we had initially thought. So we really need to dive in more and figure out what this is due to. And what we hope to work on is making this both more efficient and moving the electrons into the bacteria as well as out, and going to a CO2 fixing organisms. The other thing I want to do is leave you with a couple lessons. So this is actually a big one. We really have to express proteins rather than overexpress. When you're just doing an overexpression experiment, you don't care. You're going to kill the bacteria at the end of the day, right? Whereas for us, that's the beginning of our experiment. We really need we need happy cells to do our function, is what I think we're learning. So really balancing these two things. And the other thing I would really drive home is that the only reason we know these sorts of things is because we've spent the time to make highly quantitative measurements. And that's really been essential for unraveling these complex systems. And, cell variability matters. So since I have a couple more minutes, I'm going to quickly talk a little bit about how we're engineering cells to sculpt materials. So just to kind of give you a basic idea, what we'd like to do is trap CO2 in the forms of minerals because it's one of the safest storage forms compared to a supercritical fluid or a gas. The big problem is that if you want to do this underground, the mineralizing it is a very slow process. So it's better storage, but you're talking about thousands of years to get an appreciable amount in solid form. So, however, if you look at carbonates on the surface of the planet, most of them are from have biotic origins. So, for example, if you look at every spring in Lake Michigan, what you can see is this big cloud of white stuff appearing. And what that is actually calcium carbonate that's being formed by these cyanobacteria. Also, the white cliffs of Dover are basically skeletons of these coccolithophores. So that's all from these diatoms. So if you just globally look at calcium carbonate, most of it's been formed by organisms. Okay, and we know if we're talking about storing CO2 underground, that there are organisms under there. So even when you go 800 meters below the floor of the sea floor, you're talking about 1 million cells per milliliter still. And what's even more surprising is that if you inject CO2 underground, these organisms still survive. So they take a beating when you inject this supercritical CO2, but they'll come back. And actually, even the species that reappear are different than the ones that you start with, or they have a different distribution. So we know there are organisms that are going to be under there. We know that organisms play a critical role in mineralizing CO2. So can we actually first understand how that happens, and then can we engineer organisms to make carbonates more rapidly? So what we did is we took organisms from a wide diversity of environments, and they also have substantially diverse surfaces. So they have proteinaceous surface layers, S layers, EPS, peptidoglycan, or even lipopolysaccharide. And what we did is basically threw them into, added a saturated solution of calcium chloride to these organisms and watched how quickly you could start forming little particles of calcium carbonate. And little particles of calcium carbonate will scatter, so you can simply monitor that by the scattering at 400 nanometers. And what you see in black is if we add no organisms, basically the amount of time that it takes to lift off from this baseline is somewhere in the order of 150 minutes. Whereas if you look at all these different organisms, that happens much more rapidly. And you can actually quantitate that, and again, you see that basically compared to the abiotic case, you have several organisms that nucleate pieces of calcium carbonate up to 50 minutes faster than the abiotic case. Okay. What's also cool is that you can actually look at the surfaces of the calcite crystals after they've formed. And if you look at the abiotic case, you see these beautiful calcite crystals with nothing really on them. We also threw in silica and carboxylate-covered beads as kind of a control for these surfaces. And you don't see many of these on the surface. You'll just see a smattering. Whereas when you look at the reactions that had organisms, you just find them coating all over the surface. And actually, many of them, you can see, are entombed in the calcite. So this is telling us that the organisms are very tightly associating with the surfaces, again implicating sort of a nucleation. So the other thing that we found is that if you take calcium in solution, that it actually binds to the microbial surfaces. So microbial surfaces are negatively charged, and you can measure that by measuring their zeta potential. So that's in blue. If you add calcium here in green, you see that the charge becomes much less negative, meaning the calcium is binding to the surface. Okay. So what we were able to do is actually correlate then the charge on the bacteria with the change in nucleation time. And this is a pretty decent correlation, showing that basically as the cells become more negatively charged, the nucleation speeds up. Okay. I'm sorry. As they get more negatively charged, the nucleation speeds up. Sorry. Okay. So what we've got is this model where calcium binds to the surface of the microbes. It creates a local supersaturation, which then causes nucleation to occur more rapidly. Okay, so that's led to a very testable hypothesis. So if we change the charge, can we tune the mineralization? So what Jenny Cappucci in the lab did is basically found a system where she could tune the charge on the bacteria. She found this Colobacter vibriortis, which has an S layer which you can insert different amino acids into. And so what she did is first she took both the native S layer and then inserted binding loops to calcium into an engineered version of this. Okay. And what she found was that, first of all, this provided a way of changing the charge. So what you can see is when you put a lot of carboxylates into this S layer, the bacteria is much more negatively charged than the wild type. And likewise, if you put something that's hydrophobic into the loops, you actually end up with a less negatively charged bacteria. Okay, and we did the kinetics again, and what I'll just show you is that we've got now a very nice linear correlation between the change in the nucleation time and the zeta potential. So when you make the bacteria more negatively charged, you speed up nucleation. And this is nice because it really shows us that we have, at least in this simplified case, a quantitative understanding of how this is all working. Okay, so what we learned is that many bacteria can accelerate the formation of calcium carbonate. It's due to calcium binding on the surfaces, and we can genetically modify the organisms to tune this mineralization rate. So with that, this is perfect timing, I want to go ahead and thank my group. In particular, I want to thank Heather Jensen, who's the first brave graduate student to join my group. And as I said, she's looking out for a postdoc, and I couldn't recommend her more highly. And then Jenny Cappuccio, who has pioneered the work on the mineralization of surfaces. Heather has started handing off the baton to Behzad Rod and Matt Hepler, who's a new graduate student in the group. And Cheryl Goldbeck has been our technical staff member who's kept the entire ship afloat. And then just kind of the broader community of the Foundry has been a terrific place to do lots of great work. And then collaborators from NAB, Lars at Cornell, and funding from BES. And with that, I will thank you and take any questions. Ken.
Carol, with respect to the last topic that you talked about, one of the factors that's involved in the acidification of the oceans is the difficulty for organisms that build carbonates into exoskeletons to do that.
Right.
Is pH a factor in your
Absolutely. So pH is a big factor. So, and this is something that we really want to start tackling. So, in the subsurface, there's kind of two regimes. One is first creating a supersaturated solution, and then mineralizing from a supersaturated solution. what we've really addressed is taking a supersaturated solution and basically getting over the activation barrier to mineralize. But I think a larger time lag is basically creating that supersaturated solution of carbonates, which is directly related to the pH issue that you're talking about. It's really about how slowly can you basically dissolve silic, or how quick or how slowly you can dissolve silicates to cause the pH to jump back up. But yeah, that's dead on.
Hi, Carolyn. That was really nice work. You showed some results for a 3-electrode experiment where you had your MTR-CAB cells that looked really promising.
Yeah.
Could you comment a little bit more on what sort of current densities you can get from something like that, and what operational potential?
Yeah, so let me, so in comparison to Shionella, we're still, even with the electrochemical system, we're still about, we think, 1,000 times slower than Shionella. It's a little hard to compare because you really need to do the exact same experiment at the exact same time, but Lars and Michaela have done that on Shionella, and they said, yeah, you're down by about 1,000-fold. The interesting thing is the E. coli that we're using basically do not form biofilms. Shionella forms biofilms on the electrode. If you knock out the ability of Shionella to form biofilms, you cut the current by a factor of 1,000, more or less. So we think that basically, because right now we're basically waiting for our E. coli to drift into an electrode and knock off some electrons. So we think that's actually one of the totally critical things we have to do to get those to get the efficiency as well as the current density ups. But just to give you kind of a feeling for the best, if you take Shionella, I'm thinking you can get a couple watts per meter squared. So that's pretty decent. Sure.
Great talk. Back to the efficiency. At the beginning, you contrasted the biomass for outs and the artificial photosynthesis, and I think it was 7% versus 40%. And the approach you're taking, can you really get 40% in reality? Because there is a lot of sort of steps in between instead of direct artificial photosynthesis.
So I don't know. I guess is the best answer. So you're right. That's probably an overestimate. But and the numbers that I've seen are all over the map. So I guess if you the best that I've seen is basically people suggesting you can get 10 times more efficient than doing the biomass, which would give you 40%. But I think it's really critical on how efficiently you can get the electrons into the cells. Can you really optimize the co-transport of CO2 and electrons? But I think until someone gives you it shows that they can get 40%, then it's kind of best guesses.
I think we have time for one more question.
Yeah, you can.
With regard to the difficulty of transferring things from one cell to another, would it be possible to engineer a symbiotic pair such that conditions in one cell are ideal but different from those in the other cell and get the benefits of both optimal?
For sure. I do think, though, so I think if you're just talking about electrosynthesis in general, you need a way to when you fix CO2, you always knew you're reducing equivalents, right? So I think you still are tied to getting CO2 fixation and electricity into an organism in the same organism. Now, that doesn't have to be the organism that necessarily makes a biofuel. So you could at least offload the biofuel into another organism.
These things occur in nature.
That's right. I was just thinking about that. So there are certainly examples.
For example, produce oxygen.
Right.
They often live along with nitrogen mixing
Right.
in symbiosis.
Right.
And transfer ATP from one to the other.
To do. So yeah, I mean maybe. Yeah.
That doesn't tolerate oxygen.
Right. Right.
It has to be a different environment.
Yeah. So I guess, yeah, I mean, it's an interesting idea to think harder about, but my gut level response is you need CO2 fixation and reducing equivalents coming in the same organism, but maybe there are some tricky ways to get around that.
Thank you so much, Caroline. We enjoyed our talk. And welcome to Physical Biosciences. We appreciate your willingness to speak to us. We do have pizza. It was a little bit late, but if you'll follow your noses to the far corner of the room, it's available now. Thanks for coming.